Hello all. Today we are discussing about what is the basic contractile unit of a muscle. So in last class we have discussed about what are the structures that are what are the components of a single muscle. So today we are going to learn about learning about what are the basic components that are helping for muscular contraction. So this is the basic muscular contractile unit. So it is made up of a single sarcomere. So a single sarcomere is something which is in between two Z discs. So in between two Z discs there are many components which is made up of which is composing this sarcomere compartment. So as you can see in this, there are something called as band and zone. So as I said earlier, each sarcomere is made up of actin and myosin. So this sarcomere is a portion of myofibril that is located between two Z discs. So these are the two Z discs. In between, these are the portion which we call as a sarcomere. Then anisotropic or A band, a portion of sarcomere that extend over both the length of the thick filament and a small portion of thin filament. So this is what called as A band. So this is the thick filament and this is the thin filament. So the A band is consists of a portion of thick filament and a small portion of thin filament. Then we have isotropic or I band, which is areas that include only actin filament. So this is the part which only consists of actin filament. So this is what called as I band. Next what we have is Z zone. It's the central portion of thick filament which is not overlapping with the thin filament. So this small part is known as H zone, which is not overlapped by any thin filaments. Next is M band, the central portion of H zone, which consists of wide middle portion of thick filament. So this is what called as M filament, the central portion of H zone. So these are the basic areas in a contractile unit. So next, how the muscle contractions are happening. So the muscle contractions are basically occurring with the overlap of this thin filament and thick filament. So that's what called as overbridge interaction. So it is initiated by the arrival of nerve impulse at the motor end plate, which evokes an electric impulse or action potential that travels along the muscle fiber. We decide to contract our muscles. Basically, the neural impulse which will be traveling from our brain will be arriving to our muscle structures. So once the muscle structures receive the brain impulse or the nerve impulse, it will create an action potential. So action potential is nothing but an electrical changes which is happening inside our muscle. So whenever that electrical changes are happening, there are some chemical process which is occurring in our muscle or in this sarcomere. So there will be some chemical changes will be happening. So because of that chemical changes, the overlap will be happening or the cross bridging will be occurring. So that's the primary reason why the muscle contractions are happening. So these are the stages in a cross bridge formation. So as I said, in a single sarcomere we got both thin and thick filament. So the thin filament is called as actin and the thick filament is called as myosin filament. So the first stage when the cross bridging happening is called resting phase, where the cross bridging project from a myosin myofilament but are not coupled with the actin filament. Adenosine triphosphate ATP is attached near the head of cross bridge. Troponin covers the active site on the active myofilament and the calcium ions are stored in the sarcoplasmic reticulum. So in the resting stage, the muscle in a single sarcomere, this is your actin filament and this is the myosin filament. So in the normal stage, these are the myosin receptors which is present on the actin filament. But in resting stage, what happens? These receptors are, all of these receptors are closed by the troponin. So if this interacting crossbridge need to attach to these receptors, this troponin has to be moved from that particular place. So to move the troponin from this receptor stage, there are some chemical process happening. So you can see clearly in this picture. So this is a thin filament and this is a thick filament. So these are the bridging areas that need to be attached with this actin filament. But because of this troponin which is placed in the receptor region, the myosin filaments are not able to attach with this actin filament. So after some chemical process, this troponin will be moved from that normal receptor region and the bridging inactive cross bridges will be attaching to this receptor region and it will be producing that muscular contraction. So no need to get confused with these ATPs and sarcoplasmic reticulum. I think ATP is the basic cell which providing energy. So because of this ATP, we are able to go for muscular contractions. So ATPs are the end product from our food. So all the foods which we are having is ended in this ATP format. So next stage is coupling stage. So basically what happening in a coupling stage, this troponin will be moved from the receptor stage and the cross bridging will be attached to that active site so that it will be able to produce that muscular contraction. So that's what happening in coupling stage. So in order to remove the troponin from the receptor place, the calcium need to be attached with troponin to move that troponin from receptor site or to open that active site. So this is what happening in second stage. In third stage, what happens is once this cross bridges got attached with this receptor site, what it does is it will go for a active flexion. So that means in this you can see these cross bridges are in a elongated position. So once it attached with the receptors, it will go for a flexion movement. So it will produce shortening of muscle. So as you can see in this picture, this is the Z line. So if this Z lines are elongated, it means that the muscle in a relaxed stage. So once this thick filament got contracted and thin filament got overlapped, the distance between two Z discs will be reducing. Because of that, the muscles will be contracting. So that's what happening in the coupling stage. Or not in coupling stage, in contraction stage. And next is recharging phase. So as I said, ATPs are the one which is providing energy for each of these muscles, each of this actin and myosin to undertake some work. So as the ATP got died or as the ACP got worn out, there will be new ATPs which will be attaching to that same bridging site and the same process will be continuing. Next is relaxation phase. So once we achieve our muscle contraction, then we need to relax our muscles. So what happens in that, all the process which I have discussed now will be reversing. So the cross bridges will be retracting from that particular stage, and this receptor site will be closed by troponin again. So the muscles will be going for elongation and the muscle will go for relaxation or in a resting stage. So these are the 4 stages that are happening in a muscle contraction or in cross bridge formation. So according to this particular type, there are some muscle contractions. So first one is isometric muscle contraction. So this is the normal muscle. So this is what isometric contraction. So in isometric contraction, as I discussed earlier, isometric means the length is not changing. Length is not changing. So as you can see in this picture, the muscle is doing some work, but it's maintaining its normal length. So that's what called as isometric muscle contraction. So next one is concentric muscle contraction, where the muscles will be going for shortening. So this is what happening in concentric contraction. So next one is eccentric contraction. Eccentric contraction, as we discussed now, in usual muscle contraction, what happens is the muscle length will be reducing or it will be maintained in isometric contraction. But in eccentric contraction, the muscles will be elongating when it tries to maintain that normal muscular tension force. So these are the 3 types of muscle contractions.